Welcome to the deep dive. Today we're really getting into the weeds on the secret of life itself, the chemical nature of the gene. It's a fascinating story, actually. Involves viruses, radioactivity,
Yeah,
even a bit of architectural brilliance.
absolutely. Change biology forever.
So our mission today, pretty straightforward. We're following DNA from, back when it was a total mystery,
Hmm.
through figuring out its famous shape, and then right up to how we actually use it now, the tools. And we'll end with this amazing puzzle, how life packs this gigantic molecule into such a tiny space.
It really is the ultimate instruction manual. And, to set the stage, you got to think back to the early 20th century. Scientists kind of knew what the genetic material should do, but not what it was. I mean, if something's going to be the blueprint, it absolutely has to do three things.
Okay, what are those?
Well, first, replication. It's got to copy itself almost perfectly every single time a cell divides. That's non-negotiable.
Right, makes sense.
Second, it has to control growth and development. It's the master plan for the whole organism.
The director, basically.
Exactly. And third, this one's maybe a bit more subtle. It needs to allow for adaptation, evolution. So it has to have some way to change over time.
So copy itself, control things, and allow change.
Yep. And for ages, the main suspects were proteins and, well, nucleic acids like DNA.
Okay, so that brings us to the first big hurdle, proving it was DNA. And it started with some weird bacteria.
Yeah, bacterial transformation, that was the big early clue. This idea that harmless bacteria could somehow pick up something from dead, dangerous ones and become dangerous themselves. Griffith's experiment in 28 really showed this happening, with the smooth S and rough R streptococcus strains.
Yeah, I remember that one, the mice experiment.
That's the one.
Yeah.
And later, Avery, McLeod, and McCarty built on that. They showed if you destroyed the DNA in the mix, the transformation stopped. But if you destroyed protein, it kept going.
So pointing pretty strongly towards DNA.
Very strongly. But, science likes certainty. There was still a little bit of doubt, maybe some protein contamination. The real clincher, the sort of scientific mic drop, came in 1952, Hershey and Chase. Their experiment was just elegant.
Okay, tell us about that one. Viruses, right?
Bacteriophage T2, yeah, viruses that infect bacteria. They work by landing on the outside of the bacteria, injecting their genetic material, and leaving their protein shell, their body, outside.
So only the instructions go in.
Exactly. And the question was, are those instructions made of DNA or protein?
And this is where the radioactivity comes in, isn't it? Such a clever approach.
It really was. They needed a way to label the DNA and the protein separately, to track them. So DNA has a lot of phosphorus, but basically no sulfur. Proteins have sulfur, but very little phosphorus.
Ah, okay. So you use radioactive versions of those elements.
Precisely. They made one batch of viruses with radioactive phosphate, that's 32-PO-T, that labeled the DNA. They made another batch with radioactive sulfate 3,2-ambles. That labeled the protein code.
Got it. Labeled DNA in one group, labeled protein in the other.
Then they let each batch infect bacteria. After a bit, they used essentially a kitchen blender.
A blender? Seriously?
Yeah, to literally knock the virus bodies off the outside of the bacteria. Then they spun it all down in a centrifuge to separate the bacteria from the liquid outside.
Okay, so the heavy bacteria sink to the bottom pellet. The lighter virus coats stay in the liquid.
Right, and then they just measure where the radioactivity ended up.
Right.
If protein was the genetic stuff, the radioactive sulfur should be inside the bacteria pellet.
But it wasn't, was it?
Nope. Almost all the 3 times the protein marker stayed outside in the liquid. But the radioactive phosphorus, the PNA marker, that was found inside the bacterial cells.
Wow. So the DNA went in. That was the instruction set.
That was it. Conclusive proof. DNA carries the hereditary information needed to make new viruses. Protein is just the delivery package.
Just beautiful experimental design. And that work got the Nobel in '69, right?
It did. And once they knew it was DNA, the race was on to figure out its structure. You can't read the blueprint if you don't know how it's put together.
Right. Section 2, deciphering the blueprint.
So we know DNA is a nucleic acid, built from units called nucleotides. Think of them as the letters. Each nucleotide has 3 parts. A sugar deoxyribose in DNA, a phosphate group, and one of those nitrogen-containing bases.
A, G, C, T, adenine, guanine, cytosine, thymine.
Those are the ones. And the sugar and the phosphate link up over and over to form the backbone. It's a phosphodiester bond connecting them.
Like the sides of a ladder, right? Strong and holds it together, and it gives it a directionality.
Exactly. We talk about a 5' end and the 3' end. It matters which way it runs. But the real information, the code, is in those bases, A, G, C, and T, and how they pair up.
Which brings us to Charif, Erwin Chargaff. He found something quite simple but profound, didn't he?
He did. In the early 50s, he just analyzed the DNA from lots of different organisms, and he noticed a pattern. The amount of adenine, A, always pretty much equaled the amount of thymine, T, and the amount of guanine, G, always pretty much equaled the amount of cytosine, C.
ATGC.
Always, always. So the total purines, that's A plus G, always equals the total pyrimidine, C plus T.
So if you know the percentage of one base, you can figure out the others. Like if it's 20% guanine,
Then it has to be 20% cytosine. That's 40% total. The other 60% must be A and T, so 30% adenine and 30% thymine.
Chargaff's rules. Simple math, but it told them the bases must be pairing up somehow.
Absolutely. That was one key piece. The other was the actual shape. And for that, we need to talk about X-ray diffraction. Rosalind Franklin and Maurice Wilkins were pioneers here. Franklin especially produced incredibly clear X-ray images of DNA fibers.
Ah, the famous photo 51.
That's the one. It showed the DNA was a helix, a spiral. It also showed the helix had a constant width.
Constant width, meaning a big base had to pair with a small one.
Exactly. A purine, A or G, which are bigger double ring structures, had to pair with a pyrimidine, C or T, smaller single rings. That keeps the width the same all the way down. It also suggested the bases were tucked inside the helix, with the sugar phosphate backbone on the outside.
So Chargaff's rules, ATGC, and Franklin's X-ray data pointing to a helix with bases inside.
All came together for James Watson and Francis Crick in 1953. They built the physical model, the double helix, the icon. Two strands twisted around each other, sugar phosphate backbones outside, bases paired up in the middle.
And specifically, A pairs with T using 2 hydrogen bonds. G pairs with C using 3 hydrogen bonds.
Those hydrogen bonds, they're relatively weak, right? Easy to unzip for copying.
Exactly. Strong enough together to hold the helix stable, but weak enough individually to be pulled apart when needed.
There's one more crucial detail about the structure, isn't there? The directionality.
The anti-parallel nature. Yes, this is fundamental. The 2 strands run in opposite directions. If one strand runs 5' to 3', the other one runs 3' to 5' alongside it, like lanes on a highway going opposite ways.
Precisely. And this isn't just, a random detail. It's critical for how DNA replicates. The main enzyme that copies DNA polymerase, can only build a new strand in one direction, 5' to 3'.
Ah, okay. So because the original strands are anti-parallel, the polymerase can copy both of them at the same time, even though it only works one way.
You got it. It allows for efficient simultaneous replication of both strands during cell division. It's incredibly elegant.
Understanding that structure, that anti-parallel double helix with its specific pairing, that just blew the doors open for molecular biology, didn't it?
Completely. It led directly to understanding replication, gene expression, and eventually manipulating DNA.
Which takes us neatly into Section 3, the molecular toolkit we developed, how we learned to work with DNA.
Right. And you really have to start with PCR.
Polymerase chain reaction, Kary Mullis, Nobel Prize. This one's a total game changer, right? Fast, cheap, needs hardly any starting material.
Absolutely revolutionary. PCR is basically molecular photocopying on an exponential scale. You target a specific tiny piece of DNA you're interested in, and you make millions or even billions of copies of just that piece.
Exponentially. So like 1 copy becomes 2, 2 become 4, 4 become 8.
Exactly. After about 30 cycles, you can get over 1 billion copies from a single starting molecule. It's incredibly powerful.
So what do you actually need to do it in the tube?
Okay, you need your template DNA, obviously, the DNA you want to copy from. Then you need primers. These are short, custom-made pieces of DNA that flank the target sequence. They tell the enzyme exactly where to start and stop copying.
Like bookends for the section you want.
Perfect analogy. Then you need the enzyme itself. DNA polymerase, usually Toc polymerase, because it can withstand the high temperatures needed to separate the DNA strands in each cycle.
Toc from heat-loving bacteria.
Yep, Thermus aquaticus. And finally, you need the building blocks. The DNTPs, that's the free A's, T's, C's, and G's that the polymerase uses to build the new strands, plus salts and a buffer.
Template, primers, polymerase, DNTPs, mix them together, cycle the temperature up and down, and bam, copies.
That's the essence of it. Replication in a test tube.
Amazing. Okay, another huge tool. Restriction enzymes, RIA. These are like molecular scissors.
Exactly. They're naturally produced by bacteria. It's actually a defense mechanism.
Defense against what?
Against invading DNA, like from viruses. The bacterium's restriction enzyme recognizes specific short DNA sequences, typically 4 to 8 base pairs long in the foreign DNA, and cuts it.
It restricts the virus, hence the name.
Right. And the crucial thing for us is that each enzyme recognizes and cuts at its own unique, specific sequence. Often these sequences are palindromic. They read the same forwards and backwards on opposite strands.
So they cut very predictably.
Very predictably, at precise locations. This allows us to cut DNA into defined fragments.
And if you can cut reliably, you can start pasting things together, right? Like for genetic engineering.
Absolutely. That's the foundation of gene cloning. You use restriction enzymes to cut out a gene you're interested in, and then you use the same or compatible enzymes to cut open a plasmid.
A plasmid. That's one of those small circular bits of DNA bacteria have, used like a little shuttle.
Exactly. A cloning plasmid is engineered specifically for this. It needs 3 key features to work well.
Okay, if I want to clone a gene, what does my plasmid absolutely need?
First, a multiple cloning site, or MCS. This is a short stretch packed with recognition sites for many different restriction enzymes. Gives you options for where to cut and insert your gene.
Flexibility for pasting. Got it. What else?
Second, an origin of replication, or ORI. This sequence tells the host bacterium's machinery, hey, copy this plasmid every time you divide. Without it, the plasmid would just get lost.
So it gets replicated along with the bacteria's own DNA. Makes sense. Number 3?
A selectable marker. This is usually an antibiotic resistance gene. Why? Because getting plasmids into bacteria is inefficient. Most don't take it up.
Ah, so you grow the bacteria on antibiotics.
And only the ones that successfully took up the plasmid with its resistance gene will survive. It lets you easily select the cells that actually contain your clone gene.
MCS, ORI, selectable marker, the essential toolkit for cloning. And these tools, PCR and REs, are used everywhere now, right? Diagnostics, forensics.
Oh, absolutely. Foundational techniques.
Yeah.
But all this talk of manipulating DNA kind of brings us back to a fundamental challenge. How does the cell itself manage this enormous molecule?
Right, the packaging problem. Section 4, packaging the gigabyte. The scale here is just wild.
It really is. Think about E. coli, a simple bacterium. Its DNA is about 1.5 millimeters long if you stretched it out. But the cell itself is only about 2 micrometers long.
Millimeters versus micrometers. That's like a 1,000-fold difference in length.
At least. It has to be condensed incredibly tightly. And in humans, it's even more extreme. We have about 2 meters of DNA per cell.
2 meters. And that fits inside a nucleus that's maybe 5 or 10 micrometers across.
Yep. So how does it work? Prokaryotes, like E. coli, they do it partly through supercoiling, twisting the circular DNA up like a tangled phone cord, and then folding it into maybe 40 or 50 loops, held together by proteins and some RNA.
Supercoiling and looping.
Oh.
Okay. But for our 2 meters of DNA, that needs something more, surely.
Much more. Eukaryotes use a far more complex system involving chromatin. Chromatin isn't just DNA. It's DNA tightly associated with a whole bunch of proteins.
Primarily proteins called histones, right?
Exactly. Histones are the main packing proteins. There are 5 main types. 4 form a core and 1 acts as a linker. The core histones are H2A, H2B, H3, and H4. 2 copies of each bundled together to form an octamer like a spool.
Okay, a protein spool.
And the DNA wraps around this spool. About 147 base pairs of DNA wrapped roughly 1.7 times around each histone octamer core. This structure the DNA wraps around the core is called a nucleosome.
The beads on a string look you see in electron micrographs.
That's level 1 packaging, the nucleosome. The beads are the histone cores. The string is the DNA linking them. And the 5th histone, H1, the linker histone, helps clamp the DNA onto the octamer and organize the string.
So that already shortens the DNA considerably, but still not enough for 2 meters.
Not even close. So level 2, the string of nucleosomes itself coils up. It folds into a thicker, more compact structure called the 30-nanometer chromatin fiber.
Okay, so the beads on a string gets wound up into a thicker rope.
Essentially, yes. How exactly it coils is still debated a bit, but it achieves another significant level of compaction. But even that isn't enough for interphase chromosomes, the state DNA is usually in when the cell isn't dividing.
So there's a level 3.
Level 3 involves looping the 30-nanometer fiber. These chromatin loops, maybe 300 nanometers wide on average, seem to be attached to a central protein scaffold made of non-histone proteins.
So loops attached to a scaffold, like organizing cables on a rack.
Kind of, yeah. This creates distinct domains and further compacts the DNA into the final structure we see in the nucleus. And it gets even more condensed during cell division, forming the visible metaphase chromosomes.
Wow. Nucleosomes, then the 30-nanometer fiber, then loops on a scaffold. It's an incredible hierarchy of folding.
Molecular origami on a grand scale.
What an amazing journey. We went from figuring out what the genetic material even was,
Wow.
using those clever Hershey-Chase radioactive tags to prove it was DNA, not protein.
Right. Then deciphering its structure, the rules from Chargaff, the X-ray clues from Franklin.
Leading Watson and Crick to the anti-parallel double helix, a structure whose chemistry we then learned to exploit.
With tools like PCR for copying specific bits.
And restriction enzymes, those bacterial scissors for cutting and pasting, enabling cloning.
And finally, grappling with just how you store this immense library, the multi-level chromatin packaging with histones acting as spools.
It really covers chemistry, physics, engineering, all inside the cell.
It does. And that final point about packaging, that tight organization, leads to a really interesting question for you to think about. We've seen how incredibly dense this packaging is in eukaryotes, with all those histone layers and loops. So the question becomes, could the cell be using this packaging not just for storage, but as an active regulatory mechanism? I mean, could the very act of landing and unwinding specific regions of chromatin, opening them up or locking them down, be a primary way that the cell controls which genes are accessible and get turned on or off at any given time?
So the packaging itself influences gene expression, not just storage, but control.
Exactly. How might that extreme folding enable new layers of cellular regulation? Something to consider until our next deep dive.